So first of all, I would like to say good morning to everyone and welcome you to our today's webinar. The title of the webinar is From Research to Field, Sustainable Solutions for Potato Health. My name is 𝗙𝗲𝗍𝗂𝗍𝗶. I'm from Innocence and I'm part of the POMATO project. I'm very pleased to welcome you on behalf of all participating projects. And I want to thank you for joining us today. This webinar is organized in the context of the International Day of Potato 2026, and this year's theme is Where Potatoes Grow, Livelihoods Flourish. It reminds us that potatoes are not only an important food crop, but also a source of livelihoods, employment, resilience, and also economic opportunities across the value chain. In this context, today's webinar focuses on potato health and sustainable crop protection because supporting healthy potato production is also essential for supporting farmers, advisors, and wider agri-food systems. Today's event brings together several EU-funded projects that are all working from different perspectives on potato health, crop protection, and sustainable solutions for agriculture. The idea behind this webinar is to create a shared space where we can present different research and innovation approaches, but also we can discuss how these solutions can move closer to more practical application in the field and better support farmers and also advisors and other stakeholders. So today we are joined by representatives of 6 EU-funded projects. It is POMATO, IPMorama, NEM-EMERGE, PATAFEST, CROPSAFE, and SAGROPIA. Each speaker will briefly introduce their projects, and here you can see the agenda for today and the order of our project presentations. I will not talk a lot about these projects, about introducing them, because our speakers will provide more detail during their presentations. So we will begin with these short project presentations, followed by a joint moderated discussion, who will be moderated by Dan Milburn from 𝗙𝗿𝗀𝗃. He is a coordinator of the IPMorama project. And before we begin, I would also like to inform you, if you hadn't noticed, that today's webinar will be recorded, and this recording will be available on the POMATO YouTube channel, but it will also be shared through the participating project communication channels. So if you wish not to be shown, your camera can be turned off, and you can also maybe change your name. So I just want to inform you about the recording of this webinar. During this webinar, you're of course welcome to write your questions in the chat, and in the part where we have the questions and answers from audience, I will read the questions to all of our speakers. And to keep everything on schedule, the questions will be taken only through the chat. Before we start with the presentations, I would like, first of all, to thank all speakers and participating projects for joining us and contributing to this event. And to start, I'm very pleased to invite our Miriam 𝗙𝗿𝗄𝗍 from University of Burgos, who will present the POMATO project. And I would like to say something about Miriam. Her work focuses on polymer science, smart materials, and sensing technologies with application in agriculture, in environmental monitoring, food, and biomedical fields. So now, Miriam, I will stop sharing and you can share your presentation. The floor is yours.
Thank you, Nevena. Well, thank you very much for joining this webinar. My name is Miriam 𝗙𝗿𝗄𝗍, as Nevena said. I'm from the Universidad de Burgos, and I'm part of the coordination team of the POMATO project. Today, on the International Day of Pomato, I would like to briefly introduce how Pomato is working to protect potato crops from major bacterial diseases through more sustainable and practical solutions. Pomato is a Horizon Europe project involving 16 partners from 10 different countries from Europe and Latin America, bringing together universities, research centers, farmers associations, technology developers, and industry to move from the scientific knowledge to real agricultural practices. Potato and tomato crops are increasingly threatened by bacterial diseases that are extremely difficult to detect and manage once they become established in the field. So in pomato, we focus on two quarantine pathogens, 𝗙𝗶𝗶𝗹𝗻, responsible for potato ring rot, and Ralstonia solanacearum, which causes bacterial wilt. So these pathogens can generate severe economic losses and highly regulated, they are highly regulated in Europe because of their potential impact on agriculture and seed systems. In addition, climate change and global trade are increasing the risk of spread. So the challenge is clear. In this case, farmers are often identify the problem too late, when the symptoms are already visible and the management options become much more limited. So for this reason, Pomato aims to move from reactive control towards early detection, prevention, and more sustainable disease management with the ambition of reducing disease incidence by around 40 to 60%. So how are we approaching this challenge? Well, Pomato combines 4 different complementary strategies within an integrated pest management framework. First, we characterize the pathogen and identify resistant sources in potato and pomato varieties to better understand natural resilience against these 2 pathogens. Second, we develop early detection and surveillance tools, combining artificial intelligence models, remote sensing technologies, and also polymeric smart sensing systems to identify infections before major symptoms appear. So thirdly, we explore natural biocontrol solutions, including beneficial microorganisms, plant-derived compounds, and innovative approaches to interfere with the pathogen infections. And finally, all these developments will be integrated and validated under realistic agricultural conditions, ensuring that these conditions are not only scientifically interesting, but also they are practical and useful for farmers. Which is also very important is that all the project strongly relies on stakeholder involvement, because farmers, advisors, and also industry, they help us shape the solutions that are realistic and applicable in agricultural fields. So regarding the readiness level and the practical applicability of Pomato, it is in an earlier implemented phase because we are very young. We are only 1 year old, but we are already moving beyond concept development. During this first year, important scientific and technological foundations have been established. Pathogens characterization is already ongoing, and molecular and biological information is being transferred to support the development of early detection systems. At the same time, promising preliminary results have been observed for some biocontrol approaches, while early detection workflows, artificial intelligence supported tools, and smart polymeric sensors are already under development. The next step over the next year is to progressively integrate these developments and move towards validation under relevant agricultural conditions. And our final goal at the end of the project is to reach approximately TRL5, meaning that the technologies are tested in realistic environments and ready to support decision-making in real farming conditions. So what is the key message for farmers, advisors, and end users? Well, Pomato is not about replacing farmers' experience. It's about giving them better tools to act earlier, to make more informed decisions, and to reduce diseases risk in a more sustainable way. So our ambition is to provide practical support through the early warning infections, safer and more sustainable management strategies, and integrated recommendations that can realistically fit under current agricultural practices. So basically, POMATO is about detecting earlier, about acting smarter, and also about protecting POMATO health more sustainably. So thank you very much for your attention, and I will be happy to answer any questions you might have. And please feel free to follow POMATO project online.
Thank you so much, Miriam. Our next speaker is Dan Milburn from 𝗙𝗏𝗀𝗃, representing the IPMorama project. Dan is a plant genetist with over 25 years of experience in potato breeding, and his work focuses on genome-based breeding technologies for Ireland's national potato breeding program. Dan will also moderate the joint discussion later in the webinar, as I mentioned, but first he will introduce the IPMorama project. So Dan, the floor is yours.
Thanks, 𝗙𝗏𝗀𝗃. I'll jump right in. It's a really challenging time for late blight control in potato. You've got things like the farm to fork strategy, where the EU is aiming to reduce pesticide use and risk. Public safety concerns are accelerating the withdrawal of products. For example, in Denmark, 33 fungicides containing PFAS chemicals have been withdrawn over the last 2 years. And at the same time, Phytophthora infestans, the blight pathogen, continues to evolve. And we're seeing strains now that can overcome fungicides, including some with resistance to multiple active ingredients. And taken together, these pressures pose a real threat to potato production in Europe. So this is how we manage late blight at the moment. We use some cultural control, but we still rely heavily on fungicide applications, and that's usually supported by weather-based decisions. But you can see how this approach might be under pressure from the challenges I've just described. And one important part of the solution, and this is what IPMorama is really all about, is to bring resistant varieties squarely into this control strategy. So over the last few years, actually, numerous blight-resistant varieties have been released, but many of the same limitation in that they rely on a single resistance gene. And experience tells us that P. infestans can evolve to overcome these single R genes. And so breeders really need to develop varieties that combine 2 or 3 different resistance genes. And a major goal of IPMorama is to develop breeding lines with multiple R genes and to speed up that process using rapid breeding methods based on genetic markers. But in the meantime, we've really still also got to protect these single R gene varieties, which have enormous potential to contribute to blight control. So when we combine resistant varieties with fungicides, we really get an interesting effect, which I call kind of cross protection. And in simple terms, the resistant variety helps protect the fungicide from being overcome, and the fungicide helps protect the resistance gene in the variety from being overcome. And that gives us a basis of a more integrated, intelligent and integrated crop management system for late blight control. And in IPMorama, we're building the components of that system. So one component is what we call the virulence landscape. Now, we're really lucky in potato late blight. We have a strong monitoring system already called across Europe called the EuroBlight network. But the molecular methods we currently use to track blight don't really tell us which resistant varieties or which fungicides a blight strain can overcome. It's done more on a population genetics level. And in the IPMorama, we're creating new genotyping approaches which can be applied directly to the EuroBlight network to improve this situation. And in addition to this, instead of just reporting blight occurrences, which is what happens right now, IPMorama partners in EuroBlight have developed the Blight Tracker app, and this is a crowdsourced app that allows users, growers, extension specialists, etc., not only to identify the occurrence of blight, but also on which variety it appears. So another important component is an information resource that can feed decision support systems in which we have resistant varieties. So you just imagine map layers that I've shown here, showing the virulence landscape I just described, where potato varieties are planted in the next level and whether they carry resistance genes. And finally, weather-based risk, which means just are the conditions right for infection. And when we combine these layers, we get what IPMorama calls a vulnerability map. It's a sort of data resource that can support operational decisions about when to spray, what variety to plant, and kind of strategic level decisions about which fungicide modes of actions and resistance genes might be in threat and might have to be managed more closely. So finally, I'd kind of like to show you the potential of the approach we're developing. So last year we ran field trials using resistant varieties compared to susceptible varieties, and we compared common practice, where spray decisions are mainly based on weather conditions, with a decision support system developed by one of the partners that also takes into account whether the variety is resistant and even which resistant gene it carries. And using the DSS, spray applications were reduced by 45 to 100%. So in low pressure blight situations with resistant varieties, actually in some cases, there was no need for spraying. So even only using part of the information sort of from the vulnerability mapping that I've described today, we can already see the potential to reduce fungicide use, while also importantly protecting varietal resistance and fungicides from breakdown. And by the end of the project, we hope to demonstrate the full version of this approach, integrating all of these components at farm scale. So we also have a strong dissemination and stakeholder engagement platform as well. But in the interest of time, I invite you to visit the IPMorama website or follow us on our social media channels if you want to find out more. Thank you very much.
Thank you so much, Dan, for this presentation. And I would like to call our next speaker. It is 𝗥𝗲𝘀𝗶𝗿 from Wageningen University and Research. She will be representing the NEM-EMERGE project. 𝗥𝗲𝘀 is Associate Professor at the Laboratory of Nematology, and she is a coordinator of NEM-EMERGE project, and she will talk a bit more about this project. So 𝗥𝗲𝘀, the floor is yours.
Yeah, thank you very much. Yeah, it's a pleasure to join this webinar and present the project to the audience. NEM-EMERGE is a collaborative research and innovation project to counter the emergence and proliferation of invasive and virulent nematodes in Europe and beyond. So what is the project about? We have to face a couple of challenges related to plant parasitic nematodes, and in the project we focus on emerging nematode problems, not only in potato but also in tomato. And over the last couple of years we have seen that there are basically two important drivers that current issues with nematodes, in particular the potato cyst nematodes and the root knot nematodes. And of course, the root knot nematodes is a major issue in tomato cropping, and potato cyst nematodes is a particular problem in potato growing areas. So for the driver global warming, the issue is mainly focusing on tomato. In the southern part of Europe, also northern African regions and eastern parts, including Turkey, where we see that two main issues drive the emergence of nematodes. So one is that due to mild winters and changes in local climates, nematodes can survive, and therefore we see invasive nematode species moving upwards north. The other issue that threatens our cropping, in particular tomato, but this may be also applicable for potato, is that increasing temperatures may affect the efficacy of resistance genes. A clear example is the 𝗦1 gene in tomato that is not functional anymore above temperatures of 28 degrees. So another issue relates more to genetic host selection that drives in particular in potato issues with PCN. So after decades of applying a narrow set of resistance varieties has resulted in the emergence of virulent populations that we have to counteract nowadays in a spreading area. The same issue holds, of course, also for root knot nematodes in tomato. There we also have issues with genetic host selection. And we have defined a couple of avenues to tackle these problems. So for NEM-EMERGE, we have a couple of concepts developed on which we focus to develop tools and innovations to target these issues. And we believe that there's no single silver bullet for nematode problems. And that's also why we focus on a multiple branch approach. So for potato, the main focus is on identifying what determines virulence and how do nematodes overcome and break the existing resistance that we apply. So this is for PCN in particular, getting more knowledge on the virulence levels in fields, what determines at the molecular level also the virulence, and therefore we take a comparative genomics approach. And of course, we like in the end to match that to come up with a kind of diagnosis treatment combination where you can tell, based on diagnostic tools, what do you have in your field and what is the best response in terms of applying resistant varieties. We are not there yet, but we are making nice steps in that area. Of course, we focus also on suppressive soils, using a more general approach involving crop management to suppress basically PCN in fields like we do for root knot nematodes. We also have a concept that we work on related to understanding the molecular mechanisms that make these resistance genes non-functional at higher temperatures, and also distribution modeling for the invasion of root knot nematode species. But the key issue, indeed, as mentioned at the introduction of the webinar, is how to come from science to innovation in order to make the impact. And for that, I think key is the multi-actor consortium structure that we have. So we can join efforts together with stakeholders across different disciplines to basically move the field forward and to come up with smart and innovative solutions to target the issues that we see today. And therefore, we team up with national plant protection organizations in the project, breeders, diagnostic service providers, and several academic partners. So the consortium in total consists of 19 beneficiaries, and it's coordinated by Wageningen. And we have 16 partners from 7 European countries, but we also include associated countries that face basically the same threats, including Turkey and Kenya. And here you can see all the partners involved in the projects, and they basically represent different sectors. And by having this representation of the different sectors, we ultimately would like to have that the NEM-EMERGE outcomes and tools that we are currently developing will find their way into the sectors through our partners in the project, but also outside the project, in order to make a contribution to potato health. And with that, I'd like to thank you for your attention. And if you'd like to know more about the project, feel free to reach out through our channels in various ways. Thank you.
Thank you so much, 𝗥𝗲𝘀. And I would like to call our next speaker. It is Christian Ghideli from Funditech. He will be representing PATAFEST project. Christian is an agri-food technology researcher with more than 10 years of experience in post-harvest preservation, crop protection, and sustainable solutions for the food industry. And now, Christian, the floor is yours.
Yeah, thank you, Nevena. So, good morning, everyone, and thanks for the invitation. So, I'm going to briefly present the PATAFEST project. This project addresses emerging pest and disease threats affecting potato production and aim to develop innovative strategies for the pest management. So in the coming slide, I'm going to introduce the main challenges we are addressing, the overall objectives, and some key results obtained so far. But before the background, so as maybe all of you know, that potato is one of the most important crop worldwide. and especially in Europe. However, potato production is increasingly affected by emerging pests from abroad Europe and soil-borne pathogens, leading to important loss of yield and quality. We should also consider the presence of European entities like EPO that try to establish phytosanitary measures to prevent the spread of kind of quarantine potato pest. And at the same time, also European policies such as the Green Deal promote the reduction in the synthetic pesticide use, like creating the need for more sustainable crop protection strategies. Considering this scenario, the project mainly focused on two major categories of threat. Firstly, is a pre-harvest disease defined 𝗙𝗲𝗄𝗵. 𝗙𝗲𝗄𝗵 is generated by Candida 𝗙𝗵𝗵𝗶 soranathium and transmitted by 𝗥𝗲𝗀𝗀𝗍𝗔𝗍. This is a quarantine pest. And secondly, we are working also on soil-borne pathogen, which strongly affect the tuber quality and the post-harvest storage performance. This threat is not so an emergent pest, but consider the Green Deal and the reduction of the use of phytosanitary treatment in the field, so we can have a re-emerging of this pathogen in the post-harvest sector. So this challenge required an integrated approaches combining prevention, monitoring, resistant varieties, and sustainable crop protection tools. Some information about a number about PATAFEST Consortium. This project counts on the participation of 18 partners from 9 countries, including abroad Europe, like Ecuador, and have a strong industrial behavior, including 6 SMEs, 2 large companies, and 2 associations that are very close to potato grower and packer. Regarding the project objectives, the project combines several approaches, including the identification of resistant potato varieties, the development of bio-based plant biopesticide and post-harvest biocontrol coatings, a soil management strategy, and digital early detection tools based on imaging. And finally, also the development of VOX sensor technologies. All these solutions are being validated under real condition, both in field trials and in warehouse across different European countries and Ecuador. So the objective is not only to generate scientific knowledge, but also we would like to facilitate a practical implementation of this sustainable solution by farmers and stakeholders. Finally, some important result obtained during these 3 years of the project. We started the last year, so this June, and more than 100 novel candida-resistant genes have been achieved. Novel imaging based phenotyping system have been developed for the rapid resistance screening. The IPMorama-based classification model have been successfully developed to distinguish or to discriminate healthy potato versus potato with diseases. Regarding the sustainable crop protection, bioinsecticide solution achieve up 70% insect mortality, while the biocontrol strategies reduce the dry rot by incidence by approximately 50%. And we are now in the second year of the integrated validation trials running across Europe and in Ecuador. And from my side, that's all. So I invite you to follow up the updates of the project in X, LinkedIn, and also in YouTube. And thanks a lot for your attention.
Thank you a lot, Christian. I would like to check if our next speaker, Lorena Rangel, is here. Lorena from James Hutton Institute. If she has not joined us. Lorena? Do we have Lorena, or we can go to our next speaker? No. Okay, so our next speaker will be Bert Evenhuis from Wageningen University and Research. He will be representing the SAGROPIA project. Bert is a plant pathologist whose work focuses on crop protection strategies, integrated pest management, biological crop protection products, disease monitoring, and decision support system for healthier crops. And I would like to represent Bert. So the floor is yours.
Yes, thank you. Can you see my screen, by the way?
Not yet.
Not yet. Okay, then I will try again. Share. Should
now
be
we
okay
can
now.
see. Yeah.
Okay, thank you. So the SAGROPIA project is about finding novel pesticides, novel biological pesticides, and getting them into strategies for controlling various pests and diseases in potato cultivation. And a lot of people and organizations are working together to achieve this goal. You can see here the consortium we are working with. Well, as Dan already showed in the IPMorama case, that late blight is one of our major concerns. We are looking for organic alternatives, especially for candidates for substitution. But it's not all about late blight. It's also about early blights, Colorado beetle, wireworms, nematodes. So we are trying to find solutions for potato growers for all the problems they might encounter, looking for those organics. The first thing we did is have some greenhouse experiments in 2024 and 2025, just to see how the performance of the 12 SAGROPIA solutions we have could handle the pathogens and pests we are having. And obviously, greenhouse experiments are one thing, but in the end, you have to do the work in the field. So in 2025, we started with integrated pest management strategies in the field, and we are going to do the same in 2026. Actually, they are currently running. You can see also sugar beet here. That's the other crop we are looking in, but that's not of importance today. So the first greenhouse trials we did was on 3 different varieties with late blights. That's 𝗥𝗲𝗋𝗿𝗋 and Innovator, and we carried them out in 2024 and in 2025, and we inoculated with the EU43A1, a major strain in Europe, especially known because it has fungicide resistance against CAAs and OXTP, and therefore of interest that we find other solutions. Well, I cannot go into the details, but I can tell you that we found some very promising results. Not all of them were promising, but some of them were really promising. Obviously, in a greenhouse, you can only have monocyclic effects, so you have to go next to the field, but there's something there. As I said, we also looked there for early blights, and we again did 2 years of experiments with 2 different cultivars, 𝗥𝗲𝘀𝗁𝗿 and 𝗥𝗲𝘀𝗁𝗿 in this case. And Also here we had some promising results and some disappointing results as well. Of course, with the promising results, you can go further into the field, and with the more less promising results, we have to find other solutions. But of course, this is not about only greenhouse experiments, but this is actually also about getting that information into the field. And you can see here the experiments we did in 2025 in different countries. So this is in France, Germany, Poland, Switzerland, and the Netherlands. And in each country, we had trials on sugar beet and trials on potato, and we've in Valtemont in the northeast of the Netherlands, and we followed CAP and CAP, and we had this. We had a field trial there with 4 replicates. We had natural pests and diseases over there, and we looked at the development of the pests and diseases in the field trial. But of course, we had strategies in there, and we had actually 5 strategies. A reference strategy is just like the farm manager over there having a strategy which farmers would do similar. So that's basically your reference. Then we had SAGROPIA 1, in which we replaced candidates for substitution by organic SAGROPIA solutions, keeping in mind that we added SAGROPIA solutions sometimes to chemicals, sometimes solo. In SAGROPIA 2, we went a step further, having more SAGROPIA solutions within the strategy, and in SAGROPIA 3, we even went more and had even more SAGROPIA strategies, SAGROPIA solutions in the strategy. And then we also had a best practice strategy in there, how we would think that was the way forward. In the vicinity, we had untreated controls for early blight, corradal beetle, and late blight, just to show whether there was infection or not. Well, you can see here a picture when the field trial was laid out. And here it's maybe a bit complicated, but what we have here is those 5 strategies. And in red, you can find spray applications with candidates for substitution. In green, you find the solo SAGROPIA solutions we entered into the strategies. In yellow, the pesticides, non-CFS, and in blue, the combining strategies. And what you can see here is that in the SAGROPIA 1 solution, you can see a lot of still combinations of organic and chemical. And in C, there's more SAGROPIA solutions, solo and combis, and in D, even more. In E, you cannot see any SAGROPIA solutions because, of course, they are not registered in the Netherlands or wherever. So we cannot use them in a best practice strategy, but I really hope that we can use them sometime soon. And of course, those products could also mean something in protecting our genes in resilient potato varieties, hopefully in the future. So this is the field in early September, and you can see differences in color. And differences in color means differences in this case early blight the severity. And on the left, you can see a field which is almost brown, and that's the untreated control. And you can see that there's a lot of early blight there. A closer look 40 days later at the end of the season, and you can see one of the plots is fairly green and the rest has senescent already. With that, I would like to thank you for your attention, and these are the people who are working on this SAGROPIA project institutions. Thank you.
Thank you so much, Bert. I would like to call once again if Lorena from James Hutton Institute, she has joined us or not. Lorena? I guess that we don't have her here. Okay, so I would like to thank all speakers for their concise and insightful presentations. We will now move to the joint moderated discussion. This part of webinar will focus on some of the key challenges and opportunities related to potato health and sustainable crop protection. And I'm pleased to hand over to Dan Milburn, which already was introduced to you from 𝗙𝗏𝘀𝘁, and he is also the coordinator of the IPMorama project, and he will moderate this part of the webinar.
Thanks, Nevena. Can you all hear me? Brilliant. And thank you to the speakers for very, very clear and interesting talks. So the format we're going to take to begin with, at least, is that I have a few pre-prepared questions that I'll ask and give speakers an opportunity to answer. Some of them will be applicable to all of the speakers. Some will be applicable to only some of them. So we'll get going. And time dependent as the discussion progresses, it may become more organic, and we'll just see where it leads us and have an interesting discussion. So I think the first question I'd like to pose, and this is for each of the speakers, is across the projects, what we think, it's all very, very interesting science, and all of us, for instance, said at some point that that stakeholders are incredibly important. These are generally multi-actor projects. But what's the biggest bottleneck in terms of having a promising health solution and seeing it routinely used by growers? So basically, what do you think prevents farmers from adopting innovative, sustainable solutions? And what are the solutions in your project? For instance, in IPMorama, we're spending time to go out to stakeholders and specifically ask them what those questions in questionnaires, and then try to develop dissemination packages based on those responses. So perhaps I'd like to ask Miriam first to give us the POMATO worldview on that.
I think the biggest bottleneck in this case is the transition from the scientific proof of concept to a really good, simple and trusted field tool. Many solutions, they just work in the lab under controlled soil conditions, and the routinary adoption requires more reliability, clear protocols, also training, acceptance, also regulatory acceptance, and they have to be cost effective as well. So they need to be demonstrated that they work in the real field. So I think the key word here is the word usability. So if a solution is scientifically good but is difficult to use, farmers won't adopt it. So I think it's really important that we take all the experience of the farmers and all the stakeholders throughout the process to listen to them and to take their advice. So we listen to their needs. And for example, in POMATO, at the moment we have a survey open for different stakeholders, so they can give us their feedback. And
That's
also
great.
we are counting on them for the rest of the project through other measures as well.
Super. Yeah. So what I'm hearing is sort of demonstration of effect and cost effectiveness obviously are two really, really important things. Nevena, I see you have your hand raised, so I wanted to come back to you.
Yes, I apologize for joining the discussion part, but we have Lorena from the CROPSAFE project. So if we can just squeeze her in, and she can also present the CROPSAFE project. Is that okay with you? I'm sorry.
Absolutely.
Okay, so I would like to announce our speaker, Lorena Rangel from James Hutton Institute. She will represent the CropSafe project. She is a research plant pathologist working in the Cell and Molecular Sciences Department with expertise in plant-associated microbiomes, plant health, and sustainable approaches to disease control. And now let's hear from Lorena.
Hi, thank you. Thanks for that introduction. Let's see. Are you all able to see that? Okay.
Yes, just
all right.
Yes.
Great, thanks. Thanks for having me. I apologize. I just saw these slides about an hour ago. I'm talking on behalf of Dr. James Price, who is the work package leader for our work in CropSafe. So I just want to introduce this project. It's a relatively new project funded by the EU as part of the Circular Biobased Europe Joint Undertaking Call. So the whole remit for CropSafe is summarized really here in this quote, which isn't really a new concept at this point. It's pretty well documented that chemical pests across the globe are a threat to human health, soils, and environments. So this project brings together 14 partners from across 7 countries to address significant crop losses caused each year by pests and diseases. So this is quite an oversimplified diagram, but it's more or less a summary of our project. So we are undertaking valorized products of several waste streams. So this includes coffee waste, tree processing waste, seaweed, and fungal waste, and extracting bioactive compounds from these. We have then been testing them for bioactives against 3 key pathogens, so root knot nematodes in tomato, Fusarium wilt in banana, and potato cyst nematode in potato. So bioactives that we've found to show an effect in vitro have then been down selected for product formulation. So processing these products into a familiar agrochemical type format, for example, using granular nematicide formulation that growers are already familiar with using. So these products are then entered into greenhouse testing, where there's then further down selection, and we'll be doing 2 rounds of field trials. So here in Scotland, we'll be working with 2 seasons of potatoes. So this is going to all be coupled with the creation of pathogen modeling tools to allow for bioactive incorporation into decision support systems. And on top of all this, there's an extensive sustainability by design component of the product to ensure safety and sustainability throughout. So obviously this is the International Day of Potato, so it would be a mistake not to highlight what this means for potatoes. So a lot of the potato work that we're focusing on at the moment is quite appropriate here for in Scotland. Scotland, like the rest of the world, has potato cyst nematodes infecting its soils. Scotland also supplies the UK with about 80% of the seed potatoes that we use to grow ware. Further, 30,000 tons is exported to Europe, and an additional around 100,000 tons exported to non-EU countries, like you can see here, Egypt and Morocco. Due to EU legislation, seed potatoes can't be produced on land with PCN. So PCN infections have spread, and there's a reduction in availability for seed potato and land suitable for production. So this means a lot of our growers here are no longer able to find potato production profitable, and it's ultimately a direct threat to sustainability to potato production in Scotland, and really impacting food security globally. So this project is pretty unique in that it's really fast moving. It's moving from bioactive identification, from waste streams to field testing bioactive products in just 4 years. So it's really speeding through technology readiness levels. So the novel bio-based nematicides from these valorized waste streams, we found they're already showing some promise, certainly at the lab stage. So here in this graph, we're able to see significant reduction in potato cyst nematode hatching, which could ultimately allow potatoes to be produced with higher yields in infected fields. So these products have also been successfully formulated in tandem. This is going on with another partner into granular nematicides, and we're scaling up currently in greenhouse trials that were just set up yesterday. So once we've confirmed continued efficacy in the presence of the host plant, we'll undergo further down selections and proceed to field trials. So we'll be working on the potatoes here in Scotland. In Italy, they'll be doing the tomato work, and then our other partners working with banana will be in Tenerife. So there are good signs so far for outcomes of the project. So we're hoping, by the end of this to have some sort of marketable product support by 2028. I mean, that's quite ambitious, but that's the goal. So just thanks for listening. Here are various media links. If you want any more information about the CROPSAFE project, this is also a QR code that will bring you directly to our webpage. Thanks.
Thank you so much, Lorena. And I would like to get back to the discussion part. So we go then.
Great. Thank you, Lorena.
Thank you.
That again, really interesting, and it fits right in with the rest of the talks really nicely. We were talking just before that about bridging the gap between science and application, which I think is the single most important thing to do and the single biggest challenge. And Miriam has spoken a little bit about that, the importance of demonstration of making things cost effective and demonstrating that they're both effective and cost effective, and instilling confidence in things. I'm going to sort of open that up and see if I can get some hand raising going on here. Does anyone from any of the projects want to highlight any specific things that they think are important that they're implementing in their project in order to engage stakeholders better? And really, really build confidence in the project. Miriam, great.
Sorry, I just wanted to add that in our project, we also have field validations and feedback mechanisms. We have 4 open days planned. We have a multi-actor participatory platform, practice abstracts that the European Commission tell us to do. We have webinars and all these kinds of activities. So our message goes to more people than just the project.
Brilliant. Okay, Aska.
Yeah, I think what might be also an insight that we obtained organizing workshops together with stakeholders, local stakeholders, is that it goes both ways. So it's also a lot about awareness, also among scientists. What are the demands and how are things perceived? This could also even include the wording that you sometimes tend to use, which gives misperception. Like R gene that apparently sometimes already is connotated with genetics and then easily to GMO. So and also that it's very much context dependent. I mean, if you talk, of course, to Dutch stakeholders, you have a completely different context in also terms of cropping system than if you talk to farmers and stakeholders in Kenya or Turkey, but also within Europe. I think the levels of understanding is sometimes even more basic, where you have to maybe train and teach and target your outreach than you think of, on top of all the elements that were already mentioned by Miriam, of course, which we take care of in the project as well.
Yeah. Thank you. I think one of the things I think is very interesting is the idea. Your colleague and IPMorama partner 𝗙𝗲𝘀𝗵𝗿𝗀𝗃 once said to me, it's very hard to get a message to every potato farmer in Europe. And so one of the things we all have to engage really rigorously in this sort of workshop-based approach and demonstration farms, but you can bring a fairly limited number of people into a single site, and you're at the mercy of people's interest when you have websites and channels like that. So one of the things I think is important is to maximize our engagement with pre-existing networks that farmers are already engaging with. So, for instance, in the IPMorama project, we're very well connected to the existing Euroblight monitoring network. So I'd like to make a big ad for Euroblight. So I'd like to, and that's a very quick way, because it's already something that's very used and very embedded into the stakeholder network. We also use in a different part of the project the 𝗙𝗏𝗀 testing network, which is a part of the breeding and variety release process. Are any projects utilizing these kind of pre-existing networks to get their message out and embedding that? So I'm going to leave that open to the floor. Ashley, you're in my screen at the moment, so maybe I'll start with you.
Yeah, definitely. So I think this initiative is a great example, of course, but because we tap into the existing European project network, basically. No, but also, of course, you have partners in the project that basically also represent, for example, a society. So whether these are the European or Society of Nematologists or the European Association of Potato Research. Also EPO has been mentioned. So having your people, let's say, bridging to other existing platforms and channels is a great way to get your message across. And I think that's very valuable and could be used maybe even more.
Yeah, that's really good. I think does anyone else have? I see Bart has his hand up. That's great.
Yeah, well, we're trying to get the message across, of course, in networks. Last week we of course had the Euroblight network where both Dan and I spoke about our respective projects. But also a lot of the institutions present in our SAGROPIA project, they are also members of the 𝗙𝗵𝗹𝗶, so the biological community, I would say, which have their annual meeting usually in Switzerland. And of course, that's also a good way to meet companies who are also willing to get these products on the market, and some of our members are actually also companies. So that's one way. The other way to get messages across, and that's more difficult, is trying to do on-farm trials or demonstrations. But that proved to be a challenge for a few reasons. Regulatory, usually these projects are not registered, so you have to go to the authorities, and they are not every time willing to have these products tested at farms and being able to sell the products, of course. And for farmers, usually they are looking at these products like, yeah, they are less effective, and usually they are. And then there's the perception of risk. So we have to come up with strategies to include those products, but at the same time show the farmers that they can use them safe in certain conditions, I would say.
That's great. Christian, you have some input.
Yeah. I just want to point out just a things based on what the other colleagues say, that well, I think that when we're speaking about the stakeholder, we normally think about or the industry, well, the industry of grower, farmer, bio-based industry, we have a bio-based solution, etc. But we also have to keep in mind the citizen, okay, that are like an end user. So, and for example, for the potato crops, there's not only the higher production of warehouse, etc., but we have also urban garden owners that would like to know or to understand how the science can come to them, okay? And so just to put an example, in PATAFEST, we are focused and very focused on citizens of science activity. So our solution, for example, a biofertilizer, is tested by our partners, okay, like potato growers, etc. But also we have an activity dedicated to the citizen where they can also try and test our biofertilizer in their own small field, okay, and so provide us some feedback about how was the experience of testing these new prototypes, okay? And so this is very useful because maybe they have some issues on the application and some negative effects they may have compared to the big industry. So that's I think that it's important also to involve citizens and so bring science to the consumer and the common community, and not only scientific and in the industry.
Thank you, Christian. Okay, I'd like to move on to a sort of a different question, and that's this is all very exciting, but one thing I worry about is things are getting very, very complicated for farmers. And so if you look at the range of solutions that are being developed across projects and compare them to management practices at the moment, if you have a problem with blight, you spray. If you have a problem with nematodes, perhaps you can, if it's feasible in your jurisdiction, you can use nematicides. The problems are there and responsive, and they fit quite into management programs. And if you're a farmer now and or a grower and are looking at this and you think, oh my God, how do each of these individual processes looks quite complicated. If I have multiple problems, how do I integrate all of these things together? Because of course, potatoes don't just get blight or just get nematodes or just suffer from 𝗙𝗲𝗋𝗆. So does anyone have any sort of insights into how farmers can and growers can start to integrate all of these things together? Because one of the reasons we got together as a consortium was partly to disseminate results because it's thematically similar, but now I'm starting to see that it's also a very, very complex and scary looking thing. So how do we get past that for growers? Does anyone want to volunteer to beard the lion's den for that question? Oh, I've got a hand raised. Sheena?
Yeah, thanks. Well, as Bert already presented it nicely, in the SAGROPIA project, we are dealing with about 13 solutions or biopesticides. And if you see in his slides, in the IPM strategies in work package 3, in fact, we had all the combinations of these SAGROPIA biopesticides against cocktails of disease for potato at the same time. So maybe Aphidis is earlier or Colorado beetle is later, and the nematodes comes first before planting, and Phytophthora depending on the temperature and humidity. So what we are trying, in fact, is to have a sort of a package of IPM strategies for a potato in total. Well, that is going to be also demonstrated in work package 4, where we have the demonstration fields, as Bert has mentioned it. And I want to point out there is also a very nice strategy designed where we organize roundtables with important sector in potato. It could be producers, farmers representatives, or associations that you have, which they can disseminate this information quite nicely to the rest of the society. One thing here is, of course, the policies are not the same within the different countries. We have a little bit of challenges if it's not registered in the Netherlands, as Bert pointed it out. But last week we had a nice meeting with the Consortium in Switzerland, for instance. They are allowed to do that. So they are, in fact, inviting about 200 participants from different potato chain in order to disseminate this information which we are getting from SAGROPIA. So the issue is now just provide a sort of a package of IPM strategy for the farmer to deal with from all the spread of the disease which are dealing with in potato. Will it work practically or not? Well, Bert already mentioned it. We have very nice promising results, but getting it always into the hand of the farmer takes a little bit of time and patience.
Yeah, so I see what we, I mean, we all recognize that getting our individual solutions into the deployment landscape, as we say in IPMorama, is both a challenge, but it's the most important thing to do. But I guess what I'm really interested in to a very large extent is there's lots of solutions for different things. And so, for instance, if I look at some of the things Lorena was saying in her presentation about the application of biologicals, and they were some of the things, Lorena, you mentioned were obviously you're specifically targeting potato cyst nematodes, but I've seen some of those the things you mentioned advanced for control of other diseases as well. So, and this is a thing that's happening with some of the say with specifically with biologicals and sort of waste stream products, is that while we're testing them on individual pathogens, they may have more generic effects, and so how do we start to manage this as an issue? So you can, when you apply a blight spray, a synthetic spray, it's a simple decision. I got blight. I spray this product. I spray some 𝗙𝗵𝗵 or some 𝗙𝗲𝗹𝗱, something like that, and it does what I expect it to do, and then I can move on. Whereas if I apply a waste stream product to the ground, what am I expecting it to do, and does it have multiple effects? How do I mix it in with, if that's for nematodes, how do I mix that in with my IPM alternative product IPM control for late blight? So how do we bring this all together and get farmers through not just in utilizing our individual sets of results, but taking on board all of this information as a whole?
Well, let me give first a chance to the other colleagues before I react to that. Maybe. Yeah, thanks.
Well, I don't see any hands yet, so maybe Ms. Quiñi, you can go for it.
Well, I can give you a very nice example. For instance, the strategy that we have been using to target wireworms and nematodes at the same time in the SAGROPIA project. Unfortunately, there are two different pests, but soil dwelling, and the SAGROPIA solutions that we have didn't work for wireworm, but they work for nematodes. But at a certain point, then if we can formulate it differently, in which we are discussing with the partners, we can have also one solution for both wireworms and nematodes at the same time. Getting, as Aska mentioned, it's a single bullet, a silver bullet solution for all the diseases and pests. It's not going to be possible at all. But as a strategy, what you can do is get all the diseases and pests which are very prevalent in your environment, try to put the strategies in IPM, and provide the decision support system for that. If you have all the tools, put them in a decision support system, then the farmer can also go on for a tailor-made management system for different pests and diseases. At least this is a way that we lead in the Netherlands. I think Aska also mentioned it. Decision support system is one of the great tools that we are including in the NEM-EMERGE project. But getting one solution, imagine for all, forget for nematodes, for instance, how many do we have? Root knot nematode, potato cyst nematodes, root lesion nematodes, 𝗙𝗲𝗹𝗻, xylopholis, stem nematodes, all cocktails of rodents that we have. Do all biological control work for all nematodes? No. But we need to have a synergy or a combination of all these stuffs to bring them in available tools that we have, like the decision support system.
Yeah, so that's, I mean, I see the hopefulness and the complexity of applying these things at the same time. Bert.
Yeah, well, of course, growing potatoes is part of having a crop rotation. And within a crop rotation, you can do certain things about soil dwelling, nematodes, pests, and also diseases. So you don't always have to do something in the crop itself, but you can try to do something in between the potato crop to lower the disease pressure pest pressure. When it comes to leaf diseases, of course, then you already showed our blight app. Actually, the blight app shows you when there's a blight risk, not only for potato late blight, but also for early blight. But what it also does is having the varieties there, having the genes there which make them more resilient. It's linked to the phyto alert. So if, for instance, late blight is found on a certain variety with a certain gene, it will show on the blight app that there's disease about, and then you know, okay, there is blight, it is virulent, now I have to start applying my product. In those decision support systems, there are also products listed with which are actually allowed. Once these organic products are coming onto the market, these will also be listed in those in that product list with their characteristics. So whether it's preventative, curative, duration, rainfastness, etc., will be in that list. And as you know, uh, behind all that, it gives for each fungicide or organic, it will give you the time of the duration of your protection of your crop. And for biologicals, it might be less than for chemicals, but it will at least show you, if I apply it now, an organic product maybe protects you for 3 days, and then the blight app will show you that you are no longer protected. And if there is another infection risk coming up, then you know you have to reapply something else, or so on. So you don't need to have the farmer know everything, but you have to give the farmer the information on you need to do something.
Yeah. Yeah, I mean, I think you make a really, really great point about how it's going to have to go the direction of travel, because the series of decisions are going to be more complex. We need to support farmers in making those decisions, and the blight app, the decision support thing, which is in the farm app suite of which is the farm app suite of programs, which I think you're describing. Yeah, so it recognizes the resistance gene, it recognizes the products. It's great to see that in the future it'll also recognize the biologicals.
Hopefully.
And so then we might have a situation in which as we move through the season, if the blight season pressure is low and we think it's appropriate, but we still want to create a selective pressure to protect the R gene, there are still tools in the box to do that. So, Aska, I see you have a point you want to raise.
Well, maybe to add, and back to your original question, where are the points of conversion for tools obtained in the projects? And I think diagnostics is also a very a nice area where we probably see a lot of complementarity, because you need quite a lot of information on your pathogen system and the genetics and so on to come up with robust markers for detection. And you hear different projects working on that. Ultimately, hopefully there will be toolkits available on the market that will be applied and become available for multiple pathogens for farmers when they come with issues. So that's also another branch, I would say, where pest diseases and challenges in potato come together, but also serve then as a nucleation point for farmers to find solutions.
Yeah, so detection is as important as prevention, of course. So we need to roll those things. And I saw a couple of mentions of things like remote sensing and AI and things like that. And so these are also very, very important. And I guess you're right. There's a level of convergence where one sensor suite sitting in the field could be detecting multiple things, and that may be one of the solutions to, if we bundle all of these solutions into a some sort of massive AI system and connect it to sensors, then maybe it decomplexifies the situation for farmers. Unfortunately, as usual, AI it seems to be the time we're living in. AI seems to be the solution. Bart, you have a point.
Yeah, just a small addition to what 𝗥𝗲𝘀𝗁 is saying. Detection, yes, very important, and detection could also mean characterization of a population. For instance, if there's virulence or if there's fungicide resistance within those populations, this can also help make the farmer sensitive decisions.
Yeah. Yeah. Okay, I mean, I think we're reaching the predetermined point at which we were supposed to slip into questions from the audience. Given what we've been talking about already, I want to just give any one of the speakers an opportunity to add anything they wanted to raise that they haven't done so already. If not, then we'll with Nevena's permission, we'll advance into give the attendees some opportunities to ask questions.
Yes, okay, then we already have a couple of questions for a specific project. So we have a question from the audience from Mishgina, who asked about the POMATO project. So Miriam, are you looking resistance in wild potato accessions? Basically, are you looking for new sources of potential resistance sources?
Yeah, exactly. We are screening native and also wild potato accessions to identify the new potential sources of resistance to tolerance against Clavibacter, Cephedonicus, and Ralstonia solanacearum. So the idea is to find resistant genotypes and associated markers that could later support breeding programs and more resilient varieties. So basically, yes, we're doing it.
Thank you for the answer. We also have a question for IPMorama. What are the strategies for protecting the varieties with a single resistance genes?
Yeah, that's really important. So I call this point in time we're at the moment for blight control. You know, it's the start. I don't know if any of you know the start of the Tale of Two Cities by Charles Dickens. It says it was the best of times and it was the worst of times, and that's kind of where we are with blight control in potato, because we have all of these resistant varieties coming out. And that's really exciting because I spent my entire career trying to work on this sort of thing. But we're also in a real danger that, given the way the blight pathogen we know works, and we've had this experience before, of course, in the 50s and the mid-20th century, we released a load of things, individual R genes from Solanum demissum, and they were overcome very, very quickly as well. And so we really need to learn from the past and protect these R genes. And actually, that's the way IPMorama is structured, is to solve that problem by trying to move us on and produce these what we call these R gene stacks, which we know now are much more robust if you have 3 different resistance genes in there. For instance, it's very difficult for the blight pathogen, not impossible, but very difficult for the blight pathogen to overcome it. So what we're doing with the single R genes at the moment is very firstly is suggesting respectfully that you don't just put them out there in the field and not spray them, because that's a very bad idea. And in fact, some scientists in the Netherlands went as far as to write an open letter to suggest that that shouldn't be done. What we can do, I think, is utilize this the approach that I was talking about earlier, is that we do use fungicide products on these things, but what we try to do is do it in a way that allows you to delay the first spray. And so what you do, in fact, our genes, actually what we find is they actually quite frequently they break down in the season, an individual R gene, depending on what blight population is present. And so the trick here is to, in the first part of the season, to rely on the resistance from the resistance gene. And then here's the hard bit. Just before it breaks down, start applying your chemistry at a full rate, not a half rate, because and then what you're doing there is you're knocking back the blight, you're preventing it from, and then those strains that can overcome the resistance don't get the chance to build up. So you retain your full level of blight control, but you also, and that's giving you a little bit of protection for the chemistry, because that prevents the buildup of fungicide-resistant strains. And then you come in later in the season and do that. Now, how do we do that? That's kind of the difficult part. One of Bart actually pointed out one of the ways in which we do that is to very, very good monitoring. So when you see that resistance break somewhere else, in somebody else's field, using something like the phyto-alert app, which I didn't actually show, but it's an excellent system. The first time you see that resistance break, that feeds into your decision support, and then it says, okay, preventative spray now, because all of the things that are required for that support system to suggest that you need to spray even with a resistant variety are now present. So that's the way we're doing. That's the way we're doing it at the moment. The problem is that the resistance genes, we had an impression that they broke down. They, one was this strong, and the next one was this strong, and the next one was this strong. But it doesn't work like that. They change from season to season depending on the blight population that's present. So that's why the real key, and that's why I highlighted the importance of something like EuroBlight. The real key to this is the monitoring. And that's the monitoring that feeds into the decision making.
Thank you, Dan. We have one more question. It is for Christian. Christian stated that 70% insect mortality with bioinsecticide and separately 50% efficacy with biocontrol against dry rot. Is this field-based, and what is the variation around these figures?
Well, no, it's not field-based. These results comes from the study done in a laboratory and greenhouse. So the application of this solution then in field belongs to the validation trials that we started last year, and we are going to repeat this year. So we don't have now some solid information in order to confirm this behavior. But well, we are speaking about completely two different situations. So in laboratory greenhouse, everything is more or less controlled. Okay, so the variation, if I understand well this question, the variation comes more from the heterogeneity of potato, and so sometimes the treatment is working or not depending of the samples that you are treating. But we don't have the influence of humidity, temperatures, and other kind of abiotic, we can consider abiotic or biotic factors. Okay, so in a future webinar, we are gonna see if we can show this result and confirm or not this behavior.
Thank you, Christian. If you have, I'm talking to the audience, if you have any more questions, please feel free to write it in the chat. We still have a bit of time. Okay. I would also like to, before we continue, I would like to take a quick group screenshot for our records. If it's comfortable for you, please turn on your cameras for a few seconds. And this is, of course, completely optional. So if you're willing, please share your camera. Oh, it's great to see you all. Please smile for everyone on the cameras. Thanks. Wow, it's great to see these friendly faces. Thank you. Okay. If we have no other questions in the chat, no. I would first of all like to thank everyone who asked the questions, and thank you for all the speakers who gave their answers. Dan has something to add. Please do.
I was going to let you finish first, but Nevena,
okay.
I'd like to thank you and the POMATO project for taking the initiative to organize this day. It was, I think, really, really interesting. I look forward to this consortium doing other events like this and collaborating together for an outreach session. But most importantly, really well done, and I think it was a very, very good day. Thank you, Nevena. Thank you, POMATO.
Thank you, Dan. And I think with this webinar that we gave International Day of Potato a real spotlight by talking about potato and all the interesting activities that we have in our projects. And I would also like to sincerely thank you all speakers and participating projects from IPMorama, POMATO, NEM-EMERGE, PATAFEST, CROPSAFE, and SAGROPIA for joining this discussion and, of course, for sharing your work. I would also like to thank all participants for attending and contributing with your questions and interest. And today's webinar, as I see it, it has shown how different research and innovation approaches can contribute to a shared goal. So supporting healthier potato crops, more sustainable crop protection, and solutions that can be relevant for farmers, for advisors, and other stakeholders in practice. So after the event, this webinar recording will be uploaded to the POMATO YouTube channel. I invite all participants to follow all projects' social media, and they will be informed about any other interesting activities and events. They can also participate. And once again, thank you for joining us today, and we hope you found this webinar useful and inspiring, and we look forward to continuing this conversation. So I wish you all to have a nice day, and I wish you all goodbye. And if someone has anything to add, please do.
Thank you. Bye.
Thank you, Nevena.
Thank you.
Thank you.
Thank you very much. Bye.
Thank you.
Bye-bye.
Bye.
Bye-bye.